For question 1, as we've heard before, each object, when you observe it at metaphase, is a pair of sister chromatids. In other words, the object that I just circled could be diagrammed as a couple of double helices. Each double helix is referred to at metaphase as a sister chromatid. So that is, again, a pair of sister chromatids. In other words, that's two sister chromatids, another two, 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 and two, for a total of 16 sister chromatids. At the beginning of meiosis, there are a total of 4 copies of every chromosome in a cell. And the goal here is to figure out there are 3 different cells that are produced by meiosis in a female. The first polar body, the second polar body, and then the oocyte. So if there's 1 copy of a chromosome, like chromosome 1, in the second polar body, and 2 in the first, that leaves 1 for the oocyte, because there has to be a total of 4. So what we're looking for here is an oocyte that contains 1 copy of every chromosome. So in this case, because there are 3 out of the 4 copies in the first and the second polar bodies, we know 4 minus 3 gives us 1 left in the oocyte. An issue here is that there are 2 copies of chromosome 2 in the second polar body and 2 in the first. That's 4 out of 4, which means there are 0 copies of chromosome 2 in the oocyte in A. So A is not the answer. Let's look at part B. We have 1 copy of chromosome 1 and 2 there. So for an oocyte, that would leave 1 copy, haploid, what we want. And there are 3 copies of chromosome 2 in the first polar body, but 0 in the second polar body. That leaves us 1, which would be in the oocyte. And like chromosome 1, there's 1 copy in the second polar body, 2 in the first, which would also leave 1 in the oocyte. That is a typical haploid oocyte. Panel C, we have in the oocyte 1 copy of chromosome 1, 1 copy of chromosome 2, and 1 copy of chromosome 3. So that would again be a typical haploid oocyte. Again, here we're just summing the numbers of copies of each chromosome found in the 2 polar bodies and then subtracting that number from 4, the total number of chromosomes that are initially present in the cell at the start of meiosis. And finally, panel D, we're looking for a total of 4, and this would be acceptable, 2 and 1, 2 and 1, 2 and 1, except for this little fragment of chromosome 1, which it appears there's been a chromosome breakage or a duplication where there's too much of that part of chromosome 1. So there's 2 copies of that part of chromosome 1 in the second polar body, 2 in the first, which would leave in an oocyte 0 copies, and that would make that an oocyte that would not produce a normal diploid embryo. So in this case, the answers are B and C. For question 1, I scored this as all or nothing. It was either 16 sister chromatids or 0 points. For the bottom question, you got partial credit 2 out of 3 points if you had B or C but not both. Otherwise, it was 3 points for both B and C or 0 points. At metaphase 1, the sister chromatids line up with the capital letters. The sister chromatids the identical copies of each other, so the capital B's with capital A's, on one side of the cleavage furrow, and the other homologs on the other, the capital B's with the lowercase a's. Here. Now, here's the important part of this question. Chromosome 2 does exactly the same thing, but there could either be capital D's on one side and lowercase d's on the other, or you could draw this with the lowercase d's on the left side and the uppercase d's on the right side. It doesn't matter because these are separate chromosomes, so they can arrange either way, with capital D's on the left side or the right side. It's totally up to you. There is not one correct answer to this question. But for this response, we're going to assume that the way I've drawn it is the way you've answered it. If that's the case, then the left side of the cells will have the capital B's, capital A's, and capital D's. So we'll say capital B, capital A, capital B, capital A, and the two capital D's. So what I've done there to reinstate is that everything that was in that left cell has gone into the left of the two cells, and everything that was in the cell that was created after this division goes down here. And note the way that the chromosomes are arranged now is that the cell is going to divide between the sister chromatids, with one chromosome on one side and the other sister chromatids on the other side. So let's finish drawing this out with the capital B, lowercase a, capital B, lowercase a, sister chromatids, and the lowercase d chromatids, sister chromatids, of chromosome 2. So in this case, each of those cells produce those two cells. So we have capital B, capital A, capital D, and likewise the same in the right side cell. And then here on the bottom, those two cells become capital B, lowercase a, lowercase d, and capital B, lowercase a, lowercase d. So one possible haplotype from one of those, and you can pick whichever one you want. I hope that you indicated which one. Let's pick that one. Is one chromosome contains a capital B and a lowercase a, no punctuation between them. And when we move to discussing the genotype, the haplotype of a second chromosome, we use a semicolon, not a slash in this case, and a D. That's because, in case you don't remember, the semicolon separates different homologous chromosomes, chromosome 1, chromosome 2. If we were talking about a genotype, if we were talking about the differences between two versions of the same homolog, that's when we use a slash. That was just a reminder. Now to get back to answering the second question, what's the maximum possible number of haplotypes? Well, in this question we see one possible haplotype, which is the one that's already drawn, capital B, lowercase a with a lowercase d. But there are others as well, so I didn't draw the one that's at the top here, which is capital B, capital A with a capital D. These are the two different haplotypes that can be produced from the way that the organization of sister chromatids and the homologous chromosomes were produced here at metaphase 1. If, though, we'd put the capital D's on the right side and the lowercase d's on the left side, we would produce two different possibilities, which would be capital B, capital A with the lowercase d, and capital B, lowercase a with the uppercase d. And that's a total of 4 possible haplotypes. For this question, there were some people who earned partial credit. The most important aspect of this question was getting the alignment of chromosomes consistent from step to step. Some of you were able to arrive at the correct organization of chromatids in the gametes, but the way they were organized didn't make sense given what you had drawn either in step 3 or in step 2. That would earn partial credit. You got the right answer, but you got to it the wrong way. Also, the gamete haplotypes had to be the complete gamete haplotype, that is, the information of all of the alleles in a single gamete, and it had to have the semicolon. So some of you might have written, for example, capital B, lowercase a, lowercase d. With no semicolon, that doesn't earn the partial credit for that part of this question. Likewise, if you had just written capital B, lowercase a in this line and it ignored the contribution of the second chromosome in allele D, that also would not have earned partial credit for that part of the question. And I was lenient about the maximum possible number of haplotypes question because I didn't really specify what I meant here. So the answer was 4, as I described. But some of you who wrote 2, because you were referring specifically to the answer in step 4, where you should have only 2 different types of gamete haplotypes, I accepted that as well. To replicate the yellow parts of these chromosomes, we'll have to have primers whose 5' ends bound the yellow region. And they also have to be antiparallel. So let's finish the DNA labeling. The top strand is 3' on the right, so 5' on the left, which means the second strand is antiparallel. And that means that the sequence of primers will be here 5', and they need to be 5 nucleotides long. T base pairs with A with T, G with C, C with G, T with A. And that leaves us with a 3' hydroxyl group that can be used for DNA polymerase to synthesize the rest of that strand. And likewise, from the bottom, the 5' end starts with a T, C, G, A, C, which leaves a 3' end there for polymerase to synthesize the complementary strand to the top strand. Note that these same 2 primers will work equally well to synthesize the same sequence from the bottom 2 copies of DNA. So A, G, C, T. Oh, look at that. I forgot to add the last nucleotide on the top strand to make these 5 nucleotide long primers. So in this top example, A, G, C, T, C ends with a 3' end here, and again, a 5' TCGAC leaves a 3' end on the second homolog to synthesize those 2 strands. In this example, it wouldn't be reasonable or appropriate to design primers outside of the yellow region, because if you did, then those parts that contain the primers would be part of the DNA that was copied. That would make the yellow region wider in that case. So the primers have to be designed with the 5' ends at the borders of this yellow region. Now, the second part of this question involves a digest of this 20 nucleotide fragment, or 20 base pairs, with the enzyme ALU1, which cuts at the following sequence: 5' AGCT. And it's really critical to note here that this will not cut AGCT as shown here, which is read in the 3' to 5' direction. That won't happen with ALU1. ALU1 recognizes 5' AGCT, and there are 2 of those sites, there and there, on both of these chromosomes, both the maternal and the paternal versions. In other words, when ALU1 cuts these PCR products, it will cut, as shown in the figure, in between the G and the C of each of those. So what we have to do then is figure out, there we go, what are the sizes of these fragments of DNA and how will those appear on an agarose gel? So after digest with ALU1, we have 3 different sized fragments of 2 nucleotides, 7 nucleotides, and 11 nucleotides. And that's in the gel that's at the bottom, is that we're going to see in the with ALU, we're going to see an 11 base pair product, we'll see a 7 base pair product, and we'll see a 2 base pair product. But without the enzyme, both of these products, both the maternal and the paternal PCR amplicons, the copies of DNA that are produced, are all going to be 20 nucleotides before digest. In the absence of ALU1, there's no digest. Both of the PCR products are a single band, one product, 20 nucleotides long. But with digest, then that enzyme cuts into the 3 band sizes shown above and should leave none of the original 20 nucleotide product. In general, this was a harder question to grade because I didn't specify, which I meant to, but I didn't, that only the yellow nucleotides be copied. So many of you drew primers that were outside of the yellow region, in the flanking regions, which is totally fine because that does address the question. It would create a copy of the entire sequence of DNA, including the part in yellow, which is what I asked you to do. So that was fine. In terms of 4A then, what I was looking for were 2 primers designed to the opposite strands that had the 3' ends pointing in the correct directions to synthesize both strands of each chromosome. That was pretty straightforward. Some of you did not get that. That's okay. The most frequent error I saw here, although it was pretty rare, was either designing the primers in the opposite orientations so the 3' ends were pointing away from the yellow region and not toward it, as should be the case. That actually was the most frequent error with this question. 4B, though, provided additional concerns. One of the things I was looking for was, in the undigested well, given where you designed the primers, was it here at the 5' and 3' flanks of the yellow region, or even if you designed the primers outside of it to these regions that weren't highlighted in yellow, did you accurately calculate the length of DNA that should be generated from those primers? And the most common mistake I saw here was for students who may have drawn the primers, designed the primers outside of the yellow region, for example. The band size here in the undigested lane would not be the length of the yellow region. It would be the length between the 5' end of the 2 primers. So that was a common mistake that caused some students to lose some points or not earn some points on 4B. The next thing I was looking for, which was very subtle, so this was only worth 1 of the 3 points, was the number of bands that original band should digest into. And as you just heard, there are 2 sites of this restriction enzyme which should cut this product, either just the yellow region or the region that's flanked by primers that were designed outside of the yellow region, should cut it into 3 different pieces. So I was looking for the presence of those 3 different fragments. To identify the frequency of individuals containing allele 9 at those 3 markers, we take the product of those 3 frequencies. So, like this. And I noticed that some of you accidentally switched 0.349 and 0.394, so you should check your answer to make sure you didn't accidentally put, for example, 0.349 again in the 3rd position, but I didn't grade that down. And the sum of those 3 numbers is approximately 0.0672, or about 6.72% of individuals should have that combination of alleles. In this question, some of you noted that the information about the frequency of PFT3 allele 9 in the population was not shown in the table above. That's not the answer, though. With the information given, you can calculate what the frequency of PFT3 allele 9 is. And that's because we know that the frequency of allele 9 with all 4 STRs is what we already calculated, the frequency of the 3, which is 0.456 times 0.349 times 0.394, just as above. And if we add in this fourth allele, PFT3, then we could consider that that's a variable. So we'll call that P for PFT3, and the sum of all 4 of those will equal that number. So if we solve for P, then P is 2.87 times 10 to the minus 6. You don't have to have written that in scientific notation. You could have just used a decimal response. But that is the answer to this question. This PFT3 allele would have to be at a really, really low frequency to generate the combination of the 4 allele 9s to be that even smaller frequency shown in the question.